Thank you, Dr. Arno. Our next speaker is another person who's been at RheumNow, I think in our first year, Rich Furie, from the Feinstein Institute of Medical Research on Long Island. Dr. Furie is a leading researcher in many of the lupus trials. We asked him to talk about new concepts in systemic lupus. Dr. Furie.
Hello, I'm Richard Furie. I'm the Chief of Rheumatology at Northwell Health in New York, and I will discuss new concepts in lupus treatment. Here are my disclosures. And these are the learning objectives. Let's start with unmet needs in lupus. Now, here are the medicines. You deal with these medicines. I've highlighted the FDA-approved medicines in yellow. And the real question is, how are we doing in our lupus management with these medicines? Here's a question for you. In 1950, the 7-year survival for a patient with lupus was 50%. In the modern era, with state-of-the-art drugs that I just showed you, the mortality rate at 15 years is now, and here's a multiple choice question. It's actually about 18%. So we're still losing patients to lupus despite the advances that we have made in our lupus management. Therefore, there are major unmet needs. And if we look at mortality, so as I just said, 50% at 7 years in 1950, but now it's 18% at 15 years. So the mortality rate has certainly decreased, but less than the age standardized mortality rate. We're losing patients early on to infections and later on to cardiovascular disease. And of those aged 15 to 24, lupus as a cause of death ranked number 1 amongst all the chronic inflammatory diseases. And as far as lupus nephritis goes, our current complete renal response rate is suboptimal. It's about 40% at 1 year. And what we know is that over 40% of patients with diffuse proliferative nephritis go on to renal failure after about 15 years. And what's really upsetting is that lupus nephritis age standardized mortality rates between 2015 and 2019 actually rose. And it's much higher in blacks than in whites. Another way to look at how we're doing, and this is SLE, Dr. Kalunian will talk to you about lupus nephritis. But in this particular table, I have listed on the left-hand side various drugs that have been developed in lupus over time. And down the middle of the slide, you see the endpoint. And for the most part, it's our very complicated composite endpoints, which I'll get to in a little bit. But the point of the slide is over on the right-hand side. And what you see are the response rates in the placebo arm. But what needs to be emphasized, it's not just placebo that those patients are receiving. They're receiving standard of care. And in our lupus trials, patients come in on background immunosuppressives, so maybe 50% on immunosuppressives, about 2/3 are on antimalarials, and 3/4 are on steroids. So it's not just placebo, it's standard of care. Obviously, probably not optimized standard of care, but it's standard of care nevertheless. And what you see, or really what you don't see, is good response rate. You see response rates that are under 50%. So that means the majority of our patients in these clinical trials, and the type of patient in a clinical trial may be on average more active than in your practice, but the bottom line is that we need to do better. So unmet needs in lupus still exist. Lupus nephritis leads the list. Severe extrarenal disease is up there. We need to prevent damage, either by preventing flares or preventing damage from the drugs that we use, namely steroids. And on the list is remission induction, and we're finally talking about remission in lupus. And of course, let's not forget about the patient. quality of life needs to be addressed. All right, now I will review the goals of therapy. And these are very obvious. The tenets of therapy: do good and do no harm. But what does do good mean? It means control disease activity, prevent flares, prevent damage from the disease, and try to reduce the development of comorbidities. And do no harm refers to preventing damage from treatment, namely steroids. But it's so important, I tell our fellows and our house staff, do not treat damage with agents intended for active disease. So in some ways, there's often overprescribing of certain medicines, namely steroids, or overuse of certain medicines, namely steroids. We're always blaming steroids. We've had a love-hate relationship with steroids for a long time, since 1948, when compound D was discovered. All right, so to drive the point home, I want to show you some data from the Toronto group. And you can see the punchline at the top: activity begets damage, damage begets more damage. But the basis for saying that resides in the table. We'll start with what's in blue. And this was a cohort of patients followed in Toronto. And at entry into the cohort, you can see the SLE scores on the left-hand side. And then I have the percentage of patients who increased their damage scores over time at 1 year and at 5 years. And it's very easy to see that the more activity you came in with, namely a higher SLE score, the greater the accrual of damage. Then if you look in the middle of the table in yellow, those who came in on higher doses of prednisone, that is 7.5 milligrams or greater, had a greater accrual of damage. And at the bottom in green, those who came in already with damage, the damage index score, you can see stratified 0, 1, and greater than 1. You came in with more damage, you were more likely to develop damage over time. So activity begets damage, and damage begets more damage. And this is an iconic figure from Journal of Rheumatology back in 2003 by Daphne Gladman. And it shows you damage accrual over time. And you can see the top two contributions to damage in lupus using the damage index came from steroids, basically osteoporosis and cataracts. Now, damage is associated with mortality. And in this analysis, you can see many studies showing the same thing, that an increase in the damage score increases the mortality risk. And if you look at the bottom of the slide, you can see it numerically, a 34% increased risk of death for each 1 point increase in the SDI, our damage index. And then there are the comorbidities of lupus, and there's a lot to think about in taking care of a lupus patient. Now, just a little mention about some of the outcome measures. I'm not going to go in any detail about the SRI and the BICLA. That's kind of old hat. But let me just tell you what's on the horizon in the form of LDAS and remission. From the Asia Pacific Lupus Group came the Lupus Low Disease Activity. And the concept is an important concept because it's a state of being, and a little bit more on that in just a moment. But here you can see the definition of lupus low disease activity. It requires that the SLE score be 4 or less, and I'm not going to read the other components, but you can glance at them right now. All right, then we have a different group, the DARS group, largely European rheumatologists who've come up with a definition of remission. And you can see there are several permutations based upon clinical remission, complete remission, the clinical remission on treatment, complete remission on treatment. But the bottom line is that we're finally talking about remission in lupus. This was not in the vernacular up until a few years ago. So big difference between the concepts of SRI and BICLA versus the two that I just showed you. And I'm not sure who's credited with this statement, but I really like it. It's good to be better, but it's better to be good. So the SRI and the BICLA require a reduction in disease activity. But you can still be an SRI responder or a BICLA responder and still have a fair amount of lupus activity because they are driven by either the SLE or the BILAG, depending which one you're using. But these are indices that are accepted by regulators. Now, if you go to the right-hand side of this slide, lupus low disease activity and remission are states. So they are good to be in. They're easier to complete because basically you just have to do the SLE, but they're not yet accepted by regulators. They're certainly more stringent, so therefore you see lower response rates. And what investigators have shown, if you achieve this state, there's a lower frequency of damage accrual. So a very important point. All right, and the last part of this talk is to discuss the role of new therapeutics. I will talk about Anifrolumab and Belimumab, and in the other talk on lupus nephritis, I am sure you'll hear about Belimumab and voclosporin. Now, just a little bit of a review of the immune system. Remember, it's divided into the innate immune system and the adaptive immune system. The innate immune system being evolutionarily ancient, found not only in all animal life, but actually in all plant life as well. The responses are nonspecific, they're immediate, and there's nothing sustained about the response. Examples in humans would be toll-like receptors, the complement pathway, neutrophils. Then on the right-hand side, I have characteristics of the adaptive immune system, which is evolutionarily advanced and found in vertebrates only. The responses are antigen-specific, and there's memory to mount stronger and stronger attacks through somatic mutations. Examples being lymphocytes and antibodies, but there's clearly crosstalk between the two. So I'm going to give you examples of two drugs, one that works in the innate immune system and the other in the adaptive immune system. First, we'll talk about Anifrolumab, which is an antibody to the type 1 interferon receptor, also known as the interferon alpha receptor. And here's a schematic. What you see in the top right is the plasmacytoid dendritic cell in the center. And upon activation by nucleic acids, immune complexes, the PDC produces a lot of different pro-inflammatory molecules, chief of which are the type 1 interferons. And here's a little history of the interferon pathway. Discovered in 1957, actually, with an influenza model. So we learned that early on that it's part of our host defense against viral infections. But it was in 1979 that this article appeared in the New England Journal with the conclusion at the bottom, it is possible that the production of interferon may contribute to immunologic aberrations in autoimmune diseases. And so that led to a lot of research looking at the role of type 1 interferons in SLE. And that also then led to therapeutics. And I show side by side some of the early trials or some of the early drugs that were trialed in lupus. On the left is Belimumab, which is an antibody to alpha interferon. Now there are 5 types, or I should say 5 subtypes of type 1 interferons: alpha, beta, omega, epsilon, and kappa. And they all signal through the same receptor, the type 1 receptor. So the early study was with Belimumab, an antibody to alpha interferon, but alpha interferon only. So it left the 4 other type 1 interferons available to signal through the receptor. We did studies and got as far as phase 2 in an extrarenal lupus study, and we saw an effect with the highest dose. But we saw a much greater inhibition of the gene signature of the interferon pathway activation with the drug on the right, Anifrolumab, which is an antibody against the receptor itself and therefore blocks all 5 subtypes of the type 1 interferon family. So with Belimumab, we saw about a 25% inhibition of the gene signature. With Anifrolumab, about 90% inhibition of the gene signature. And that explained the better clinical response with Anifrolumab compared to Belimumab. So I'm going through about 10 to 12 years of clinical research and just showing you the top line data from the phase 3 lupus trials with Anifrolumab. They were called the TULIP trials, and you see TULIP 1 and TULIP 2. And to the absolute shock of the lupus community, TULIP 1 failed to reach the primary endpoint of SRI. But TULIP 2, the endpoint was changed to BICLA prior to unblinding of the data. And we saw a positive outcome, not only actually with BICLA, but we saw a positive endpoint response with SRI. And then I show you some of the secondary endpoints. So if you followed the lupus clinical trial adventures over the last 25 years, you know that we have struggled. And so there was a lot of anxiety about the failed, or at least quote failed, unquote, TULIP 1 study. But in the end, the totality of the data were appreciated and the drug was approved. So I'm going to just show you a little bit of the data from the TULIP 2 study and then some of the pooled data. And here, this particular graph tells us two things. And in order to be a responder in this analysis, one had to achieve a BICLA response, which was the primary endpoint in TULIP 2, and sustain it until the study end. So this speaks to efficacy, but it also speaks to durability. And if you look in the little box here, you see the hazard ratio is 1.55. So that means those people who received Anifrolumab had a 55% greater chance of being a responder in this analysis. Another analysis, and we actually saw this back in phase 2, and that's in fact what this particular slide is from, that the skin responses were very dramatic. We studied 2 different doses compared to placebo in gray, the 2 doses in red and blue. And you can look at the table for the actual data. But the bottom line is that Anifrolumab worked very nicely in those with very active skin disease. And it's no surprise because PDCs, plasmacytoid dendritic cells, are very abundant in the skin of lupus patients. They make a lot of interferon. The 2 studies, TULIP 1 and TULIP 2, focused on the 300 milligram dose, and therefore it was possible, since we did very similar capture of information from the patients, to pool the data to increase the power of our analyses. And in this particular publication, you see baseline characteristics listed on the left-hand side. And if you're not used to looking at forest plots, you can see down the center a forest plot, and I'll tell you there are 3 components of a forest plot. You have the horizontal line, and that represents the confidence intervals, so they're anchored. And then you have the dot, which represents the point estimate. And then crucial is the vertical dotted line, which is, I guess you could refer to it as a line of no effect. And regardless of the different baseline characteristic, and you can see interferon gene signature at screening, age, sex, BMI, race, and it will continue on to the next slide, the point estimates all favored Anifrolumab. You can look at the bottom to see the key. Favors Anifrolumab is to the right of the vertical dotted line, and to the left favors placebo. Now, some of the confidence intervals cross the line of no effect, in which it would be considered a null effect. But that's largely because the subgroups were very small. So you have to be careful in trying to interpret subgroup analyses. And this continues on to look at region and onset of disease, the baseline corticosteroid dose, SLE scores at entry, whether they're very high or relatively low, and serologic markers. But anyway, all the point estimates, as you can see, favor Anifrolumab. Another key thing in the management of patients is to try to taper steroids. But the problem is when we taper steroids or immunosuppressives, we worry about a patient flaring. But in this analysis, and this is of the combined TULIP 1 and TULIP 2, it shows that patients were more likely to be able to taper steroids and remain flare-free. So the y-axis, percentage of patients able to sustain a steroid taper to less than 7.5 milligrams per day if they started out at 10 milligrams or more, and they were able to sustain that for the last 12 weeks of the study without flaring. So about 2.5-fold increase in those who were able to achieve this if they received Anifrolumab. All right, and let's finish up with B-cell directed therapies. There are a lot of targets on B cells, but the focus right now is on targeting BLyS, B lymphocyte stimulator. And there are different ways to do that. I'm just going to talk about the monoclonal antibody Belimumab. But you can see that BLyS and APRIL bind to key receptors on B cells, BLyS receptor, TACI, and BCMA. So this is an antibody against BLyS, which is also known as B-cell activating factor, or BAFF. And I don't think I need to review the top-line data. These are old hat. This is from 2011, but these are the two studies that led to the approval of Belimumab for SLE. But a question that came up a lot after its approval was, who should I use this on? What domains does it work on? This was a post hoc analysis of the combined BLyS52 and BLyS76 trials, and you can see highlighted in yellow across the bottom are the SLE domains where statistical significance was seen, and those included skin, joints, immunology, and on the right-hand side you see vascular and CNS, but there weren't that many patients who came into the study with activity in those areas. And this was analysis of long-term extension data. Phase 2 and Phase 3 both had long-term extensions. They were all open label, so you have to be a little careful because there's no comparator and therefore difficult to interpret. But on this particular slide, you see components of the SRI. Well, you see in the top left the SRI itself, and then in the top right, the SLE score. Bottom left is BILAG worsening, and on the right-hand side in the bottom, physician global assessment worsening. And basically, what this tells us is about durability of the response over a lot of years. These extensions went for somewhere around 8 or 9 years. But again, they were open label. They did provide safety. There were really no new safety signals over time. But another question, no one understood what an SRI response was. And so an analysis that we did was to combine BLyS52 and BLyS76 data, and we stratified by SRI response, responders versus non-responders. And this was done irrespective of treatment allocation. So we didn't care if they got Belimumab or placebo. We just wanted to know, what does it mean in everyday language to be an SRI responder? And here you see a table that shows just that. We have laboratory and clinical correlates. On the left-hand side here, you see the SRI response, and on the far right, the SRI non-responders. Again, this is agnostic to treatment assignment. And so the bottom line is, if you did well, you did well. And I could focus on some of these. These are in your handout. You can read them in greater detail. But we do worry a lot about severe flares in our lupus patients, but you can see there was about a 5-fold reduction in the severe flare rate. We also, as I mentioned earlier on, have a love-hate relationship with prednisone. So those who were SRI responders were more likely to be able to reduce their prednisone to under 7.5, and they were far less likely to have to increase their prednisone. There was serologic improvement in the SRI responders. And then as far as PROs, at the bottom of the slide, you can see that there was greater advantage to being an SRI responder with respect to SF36, with respect to fatigue. And damage. Damage is very hard to measure in a 1-year trial, or for that matter, a 2-year trial. So this was a rather interesting study published by the Toronto group by Murray Urowitz and his crew. And it compared damage accrual rates in his cohort in those who did not receive belimumab over time versus those who were in the extension study, the phase 2 extension study, who obviously were receiving belimumab. Now you might say, well, it's apples and oranges, different patients and so forth, but they went to a lot of effort to try to match patients using propensity score matching. And if you look at the left-hand slide, I have some of the numbers over there. And those numbers represent the 5-year change in the SDI, the damage index score. And the short of it is that you can see those on belimumab had a less of a reduction in the damage accrual compared to the cohort in Toronto. So not direct evidence, but as close as we could probably get to showing that damage may be lessened, or damage accrual may be lessened in those who are on belimumab. So just to sum up, learnings about anifrolumab, though there were inconsistent SRI results, there were very much consistent and robust BICLA results across the 2 TULIP studies. The safety was quite good, though a higher frequency of shingles. And so when I put patients on anifrolumab, I require them to at least get 1 injection of Shingrix. There's limited information about COVID, but there is some information gleaned from the long-term extension trial. We know, at least looking at the kinetics of response for some of the domains, especially the skin, that the drug works very rapidly. And as I showed you, there's benefits across many different domains, demographics, and baseline characteristics. The efficacy was sustained. Not only does it reduce flare rates, but it allows patients to taper steroids and also prevents flares at the same time. Now, I didn't show you the data, but we did a similar analysis to what I showed you with belimumab and looked at BICLA response, again, agnostic to treatment assignment, and how that corresponds to goals of clinical care. But as you can imagine, if you're a BICLA responder, that's better than being a BICLA non-responder. There's a lupus nephritis trial, a phase 2 study that was performed, and there was benefit seen in the higher of the 2 doses, but that study is being repeated in a phase 3 study. And as I mentioned, there is a long-term placebo-controlled extension trial, so it's pretty unique in that it was placebo-controlled, and that provided safety data and a bit of efficacy data. And lastly, learnings about belimumab. We have seen benefits in lupus as well as lupus nephritis, and there have been consistent results across multiple studies. And I don't remember exactly how many studies have been done after Bliss 52 and Bliss 76. It's probably around 5 or 6, but they all reached statistical significance with the exception of the Embrace trial, which fell just a little bit short. That was a FDA mandated study, a post-marketing study in African Americans. The safety profile is very good with belimumab. It may take a little bit longer to see an effect, but we did see effects in mucocutaneous, musculoskeletal, and immunologic domains. The efficacy seems quite durable, and as I showed you, the SRI response corresponds to our goals of clinical care. And then as far as flare prevention, we have shown that, and we also showed in the Bliss LN trial that there's a blunting of a reduction in the GFR. So again, it may speak to damage prevention in lupus nephritis. So there's no question the future is bright for our patients with lupus. We're seeing a lot of activity in drug development. So imagine a future when patients come for appointments and take their medications. Now, I don't know about you, but I have a lot of non-compliant patients and non-adherent patients, and I show you this slide. The hydroxychloroquine level is far subtherapeutic in a patient who's alleging that they take hydroxychloroquine 2 pills every day. This is a big problem, and I don't know how to solve that problem. But you probably thought I was going to come up with some novel therapy. But it would be great just to get patients to show for appointments and to take their medications, and that could potentially give us an effect size that we kill for in a clinical trial. So I thank you very much for listening.